Hi everyone, so in this video we'll talk about DNA sequencing. So let's get started. So the first method for DNA sequencing which we'll be talking about is chemical degradation method. It is also called as Maxim-Gilbert method. So in this case, a purified DNA will be sequenced and a single stranded DNA with radio labeled with gamma 32 phosphate will be used for sequencing, and it is done with treatment of phosphatase and kinase. So I'll explain you the entire method of chemical degradation method, which is the first method in this video for DNA sequencing. So this is the overall method or the protocol for chemical degradation method. So the labeled DNA fragment is divided into 4 aliquots. So in this, a single stranded DNA is used which is radio labeled with gamma 32 phosphate. So how we are going to use this particular single strand DNA? So we are going to divide it into 4 different types of test tubes or aliquots. So aliquot 1 would contain or would be treated with dimethyl sulfate at alkaline phosphate, which methylates G base. So this particular test tube, this particular aliquot will be treated with dimethyl sulfate, which will be at an alkaline pH of 8, which will methylate the G base. So this will act upon the G base and try to remove those G base. Coming to the second aliquot, which will be treated with piperidine formate, which is at pH 2, which will weaken the glycosidic bond of adenine and guanine by protonating nitrogen atoms. So this particular test tube or this particular aliquot will remove the adenine and the guanine bases. Coming to the aliquot 3, which will be treated with hydrazine, which will modify C and T bases. So this particular aliquot will modify or try to change C and T bases. And coming to the last aliquot, which will be treated with hydrazine in the presence of 1.5 molar NaCl, this particular aliquot will modify only base C. So there are 4 different types of aliquots that are used in the chemical degradation method for modifying or removing these bases for DNA sequencing. So the first aliquot will be for G base. The second is for, we have A plus G. The third is for C plus T, and the last aliquot is for C. So this is how this particular DNA, let's say this single stranded DNA, will be divided into. So moving with the pictorial view, so you can see a pictorial view on your screen, which is a single stranded DNA which is radio labeled with 32 phosphate, and these are divided into 4 different aliquots. So this is the first aliquot, this is the second aliquot, talking about the third aliquot, and this is the last aliquot. So C base was the last aliquot, pardon. So this is the C1 is the last aliquot, C plus T is the third last aliquot, and G is the first aliquot, and A plus G is the second aliquot. So this is what we discussed. This was the protocol for the chemical degradation method. So this is how it will be done. So talking about the first aliquot, which is the G aliquot. So this particular aliquot will try to remove the G bases in the single strand DNA. So in this you can see it will take only those bases, or this particular aliquot will take only particular sequences which do not contain G bases. So you can see this is the starting material, which is this. So this is the C, so I'll write it boldly for you. So this is G, C, T, A, then we have C, G, T, A as well. So this is C, G, T, A and C, G, T, A. So it will stop when it surpasses the G base. So it will start off in the right direction. This will start off in the right direction, and as soon as it encounters the G base, it will stop. So you can see at this point of time, so this is a G base, right? So this is CGTA, CGTA. So this is G base. So when it encounters G base, it will stop here, and the rest it will take. So the rest base, which is GCTAC, it will take. So you can see here it's written GCTAC, which it will consider, leaving all the rest behind. So it will stop. So as soon as it encounters a G base, it will stop. The same logic goes for all the 4 aliquots present here. So this was about G base. So talking about the second aliquot, which is A plus G. So in this particular thing, it will stop as soon as it encounters a base or a G base. So any of the two above. So I'll again write the sequence for you, which is GCTA, then we have GCTA, CGTA. So it will start off in the right direction as discussed. So this logic will be the same. So it will start in the right direction, and we see the first base is A. So it will exclude A and it will take all other. Alright, so it will take all other, which is GCTA, CT, CGT. But we will just check on for more other bases if it contains any more A or G base. So it will take this particular thing, this particular sequence, and it will keep on scanning for A or G base. So as we move here, we see another A base here, but before that we also have a G base here. But so what it will do now, it will scan till here. So it will remove this part as well, and it will take another sequence, which is this part. So now we have GCTAC. So this is the third sequence it is taking. So it's scanning. So it will keep on scanning until and unless no A or G base is present in the DNA sequence. So we started off by removing the first base, which was A base. So it did not require A base, so it removed A base and it took the rest over sequence. Then it scanned again and it found a G base. So it removed that G base and took the rest over sequence. So now we have this particular sequence which is left with us. So now it will again scan, and we have found again a base here. So it will remove the this particular part as soon as it encounters a base, and the rest it will take, which is GCT. So you can see here we have 3 sequences here. So it will keep on scanning. So as soon as we scan, as soon as it scans, the rest sequence it will be taking, and the rest sequence it has, it will go on scanning again. So it will keep on scanning and scanning to remove all the A and the G bases. So you can see the first sequence it takes after removing the A base. So it goes under scanning again. Then it finds a G base and removes the G base to find another sequence. And this particular sequence goes on scanning again. It finds a base. So it removes the A base and we get this final sequence. So we'll take all the 3 sequences, right? So this is the method of chemical degradation. We keep on degrading in a stepwise procedure. We do not do in a single process, but we do in a stepwise procedure. So same goes for C as well as C and T aliquots. So this is how we'll find out all the sequences. So this is how we'll find out all the sequences this way, and we'll plot it in an agarose gel. So this is a sequencing gel that we are using, that we'll use, and we'll order it like this. We'll order it in the reverse direction of the DNA strand which is available to us. So the single stranded DNA that is used as a starting material for chemical synthesis will use that particular sequence here. So we'll use this sequence. So this is what we have here. Alright, so this is the one that we have here, which is GCTA, CGTA. So we are using it in the right hand side, so from the right hand order. So the order is GCTA and CTA. So we are using from a right hand order, which is ATG CATCG. So we are using in the right hand order to make sure this is this. Alright, we are not counting G because G is the 0th or the 1th base. So it's not of much importance, the first base, but we are taking in the right hand. We are taking the reverse order to do this. So according to the size of these DNA sequences, we are plotting the bands here. So you can see here for the first one, let's say for a plus g. So a plus g, we have 3 sequences. So the sequence length for the first is 1 2 3 4 5 6 7. So 7 comes here. Then we have another second basis, 5 nucleotide bases. So we plot it again, 5 here. Then the lastly we have a 3 base, which is 3 here. So this is how we are plotting it, and this is what you need to show whenever you are given about or talk about chemical degradation, and show in a broader manner. So this is how it's done, and it starts from a single stranded DNA, and this is how you're gonna plot about. So whenever you get a single strand DNA, so let's say you get a single stranded DNA structure, let's say ACGT. So you'll write it in the reverse order. So you will write it like TGCA. So it will start from the right hand side. Alright, so this is how you're gonna plot on the graph paper, and also the markings for the numbering will be in the higher to lower manner, or you can start from 1 2 3. So we can start from here to go to the higher end, which can be 7 8 9 10, anything. So then this is how you're going to plot about things. So this is the same we are doing for G as well, we are doing for C, and as well as for C and T. So let's just keep this video till here. Hope you enjoyed this video, and thanks for watching.